So today, here to invite you to explore a little bit on trajectory inference analysis. So I'm Paulo Czarnewski, and I'm working now at my own company, Precisium AI. So let's dive into the trajectory inference analysis. So right now in your workshop, you have probably already gone through, you have the reads, done some quantification, quality control, normalization, feature selection, scaling and regression of your data, data integration, and then done some graph construction or visualization, for example, using UMAP, graph construction and integration also using Harmony. And from that, you'll do cell clustering, and you already probably just finished doing differential expression analysis and then identification of the cell types. But one thing that you can do in addition to that, and that's where trajectory comes, is ordering your cells based on some developmental time, and then doing differential expression on that cell ordering. And that's basically what we are covering now in this lecture. So what is the trajectory analysis trying to do? It's trying to establish some sort of ordering of your cells, and that ordering usually is named pseudotime. So it's an abstract definition of the progress of certain development. So you imagine that when your cells are activated, not all cells, so for example, here in this figure here, you can see experimental time, so cells that were incubated, for example, in a well at different time points. As you can see, all cells, they start developing their process in activation. It can be differentiation, maturation, in independent manner. So one of the key important things to distinguish is that your experimental time is somewhat different to your pseudotime. So the pseudotime is measuring within each cell where it is in this process of maturation, activation, development. While your experimental time sometimes might be related, as you can see, many activated cells are there, but this is different from the pseudotime. And that's a very key important difference that you need to consider or think, because the pseudotime is more on the genetic development rather than on temporal itself. So also keep always in mind that when cells are differentiating, as we can see here, they are all usually independent of each other. So you can see even in the most time point here, the most differential time point, not all cells will be differentiated. So, and that is actually what you will see in single cell data analysis. So there will be some biases based on where each cell is. There are a lot of different methods for trajectory inference, and we'll go a little bit more into details on those as well, and a bit of the biology and how you can see the trajectory methods being applied. As you can see, there are many different methods, and in this single cell RNA-seq tools, they have a compilation of like when some methods were developed and so on. So and you can see that it grows exponentially. But one thing that you can actually notice is that most of them are very similar to each other, even though there is a lot of methods being developed. Most of them are somewhat following the same structure. And I'll mention a few of those later today as well. One of the most important things when choosing a trajectory method to use is trying to identify which trajectory types are there in your data set. So here I'm just exemplifying a few. So for example, you have cell cycle. So just as cell division, cell proliferation, circadian rhythm, they are always a cyclical pattern, or activation, deactivation cycles in some cell states. So it's a process that is usually represented by a cycle. There are the linear trajectory type, which is usually just events that succeed each other without any bifurcation. So for example, if you have the embryo, like the cell oocyte, and then you transform that to a fertilized embryo, and then you have a stage of 2 cells, 4 cells, 8 cells, and so on and so forth. That is a very linear trajectory. There is no other things that happen in between. Once you have things that happen, so for example, if you have a bifurcation, so like this cell type can differentiate into cell A or cell B, then it comes on the bifurcation part, or it can even be differentiated in multiple bifurcations, or like a succeeded bifurcation or multifurcation strategy. So for example, most of the immune cell differentiation processes or for differentiation of epithelial cells in the intestine. They usually are divided in this category. So if you have a cell, and then you generate a progenitor of, for example, secretory cells in the intestine, and from that you generate a few of them, and then from those there are other progenitors, for example, enteroendocrine, that also generate the mature endocrine cells. So one thing that you can actually notice is that these ones, for example, tree multifurcation, are simply just a combination of many bifurcation trees coupled together. And I'll come to that in a few slides as well. So sometimes it is easier also to do trajectory analysis on parts of the biology. So for example, I want to study only the enteroendocrine cells. So you can actually just try to focus on that one instead of taking all cells in your data set into consideration. Similarly, so for example, here is another like more complex graph. So for example, a connected graph that you have the pathways, and then you have bifurcations, and even adding cycles on top. So you will see, I will show you a few examples of that as well. But these cycles can be still a combination of cell proliferation with the differentiation path. And this is very common actually in stem cell niches. And I'll show you one example there. And finally, the most complex of them is a disconnected graph. That is, for example, when you have different cell types. So if you want to model everything at once, and that becomes very, very complicated. So you have epithelial cell differentiation in here, and here you have fibroblast differentiation. They are completely different cell types, and that's why there's no connection between them here. But there are a few tools that allow you to try to model all of them at once. Let me show you, before we go to the methods and so on, just a few examples of these biological trajectories and where they are happening. So one of the things that I will always try to remind you is that, can you split in trajectory into smaller and simple trajectories? So when you're doing the trajectory analysis later on, what you can do is, so you can always take, for example, you have epithelial, or no, not epithelial, sorry, intestinal tissue that you run single cell. You don't need to run trajectory on all the cells that are in there. You can just simply select, I want to select only the epithelial cells and run trajectory on those. And then you take only the fibroblasts and run trajectory on those. So feel free to subset your data set to perform trajectory analysis as it makes sense to you, or what is important on your biological question. So for example, here is just one example. You have intestinal stem cells that they differentiate into these trans-amplify, and then they start proliferating a lot. So this is actually a cell type that performs cell cycle. And then it differentiates into absorptive progenitors, and then the mature enterocytes, or differentiate into secretory progenitors, and then differentiate into other secretory means epithelial cells. So you can see, like, if you only want to model this part here, you could in theory just choose the linear. Or if you want to model the stem cells until the absorptive progenitors here, you can then maybe choose a bifurcation, for example. Or if you want to model the whole process, maybe either a tree or a connected graph. So feel free to choose the method or the complexity that you want to explore in whatever biological system you have. Now, for example, for the skin, it's exactly the same. So depending, for example, when you have just a normal skin without any secretory glandules, the process is pretty straightforward. So the stem cells start proliferating here, generate this stratus spinosum, then the cells start differentiating to granulosum and being covered, and then these cells usually die and form the stratocorneum. As you can see, for example, in this schematic here, is the epithelial cells. They start proliferating and start generating these in a very linear fashion. If you're only interested in this part, basically a linear trajectory with all these cells here would suffice. But then if you have like a skin that has, for example, hair follicles, glands, and other types of tissues, this becomes a little bit more complex. So then you probably will need to choose a trajectory method that allows you to do that, such as trees or connected graphs. Also, that works also for different species, not only humans and mouse, but if you're working with parasites, like in this case is Toxoplasma gondii infection, you can see that cell cycle is also captured there. And then in this case, there's not much to do, especially in this data itself, they were primarily interested in exploring the cell cycle within toxoplasma and how that is being changed. And for that, it's clearly that you should use a cyclic differential expression analysis. In the exercises today, you will see a differentiation path within the immune sites in the bone marrow from the stem progenitors to all the mature cell types. As you can see, also depending on which type of cell, so for example, maybe you're only interested in lymphocyte or T or B cells, for example, then you can choose maybe a linear trajectory from the start, and you just subset your data set and use only those cells and define a linear trajectory. Or you can try to explore all of them, and that's actually what we'll do in the exercises a bit. So basically, we created a tree for this data set, and then you can explore them as a whole, like the whole thing at once. Also, for example, embryonic development, that's what I was mentioning. Like you have a fertilization, the oocyte, and then it transforms into the first embryo, and then the 2-stage cell, 4-stage cell, 8-stage cell. This is a very linear trajectory. So if you're only studying these parts here, then a linear trajectory will suffice. But as you start developing after that, so if you go to blastula or to gastrula, and the development of the notochord and the other parts of the embryo, then it starts becoming more complex. So maybe a tree or a connected graph if you want to study the whole thing, or if you want to study this part specifically, then these more connected graphs will be better suited for that. As a last example, so here is also an example where you have stem cell progenitors. This is a data set where there were stem cell from the neural development in the embryo. So it's the first stem cells that will start generating the neurons that will populate in the brain and the spinal cord. As you can see here, in different developmental ages, you can see the different shifts in proportions. So for example, in the beginning, you have a lot of these cell types in here, the green, and then that starts shifting and transitioning to more and more neurons, as you can also see in this graph. But one thing that is very important to see here is that there is these loops around here, and you can also see that in the figure here, these arrows. It's the arrows of transitioning of the cell cycle. So these are actually different cell types or different progenitors that they cycle around, and then they at some point they exit this loop and then start differentiating into the mature cell types. And that's a very clear concept for this connected graph. So you have a progenitor cell, and then the stem cells, they keep this cycling loop to generate more and more progenitor cells. And some of those will then exit that cell cycle phase and start into the differentiation and final maturation into mature neurons. And those later will remain in your body for the rest of your life. So for this type, trying to study this whole process, then maybe a connected graph would be a better suited alternative here. So if you have any questions, please, I'll give you 5 seconds to raise your hand if you have any questions until now.
I mean, I'm curious how important it is to really nail down the exact like trajectory model that we want to use.
Yeah, it's very important because actually you will choose the tool that you want to use based on that. Because not all tools are suitable for all these applications. There are tools that only can apply to one or another. So for example, as you get more complex, the number of tools that you can use is actually very limited. So there are maybe 2 tools that you can use. So that's why I'm always saying also to try to simplify the trajectories, because sometimes, for example, even though you have the cell cycle here, maybe what you're interested in is, actually, do you see my mouse? I'm pointing things.
Yes.
Okay, good. I should probably just change it to this one. Maybe it's a bit better. So what you can do is also just subset this part of the data set. So for example, if you're only interested in differentiation of the neurons, you can simply subset this part and apply a simple method that usually there are many packages that allow you to do that. So, and you will see actually on my last slide, I have a summary of these types of trajectory topologies is what these are called, and which methods are suitable for some of those. So for example, I think for our lecture and the exercises, Paga can actually take care of this one, not much on the disconnected, a little bit, but it's really hard to use actually. And Slingshot, which is the other one, can reach up to this one. So these are kind of in level of complexity, but and trees and connected graphs are the ones that usually you will see quite often.
Thanks.
Thank you for your question. If no other question, then I can go a little bit into how trajectory inference methods actually work. In essence, there are hundreds, as I just mentioned, there are hundreds of trajectory methods out there, but basically most of them will follow some sort of decision process. So for example, how do you compute the similarity between the cells? So how do I know that this cell comes next to another one? So one of the ways that you can actually try to measure is also by looking at the manifold. So like, are you using the PCA embeddings or the harmonized embeddings or the UMAP or the ICA, PCA? There are different embeddings that you can use. And as a general guideline, you should try always to use the methods that have more dimensions than UMAP or TSNE. TSNE and UMAP are good for visualization of your trajectories, but usually you should use, for example, either the harmony, like the harmonized embeddings that usually have removed batch effects, or for example, diffusion maps or other types of embeddings that are multidimensional, not only two-dimensional. Following that, you'll do clustering, which you already have done. So even the embedding, you already have the harmony embedding. So this is already done, or whatever scanorama embedding and so on. So you already have done that part. You can then choose the clustering. So for example, you could use the Seurat clusters or the clusters from the graph clustering on ScanPy that I don't know it's still Louvain, right? I think or Leiden.
Leiden.
Exactly. So use those clusterings, and then from those you can then create your graph, your connecting graphs. So for example, which subclusters are connected to each other or are close to each other in space, and that's primarily via what you call the MST, minimum spanning tree, which is my next slide, and I'm explaining that part. And from that, you can then try to identify which cluster is the one that is your stem cell. Usually that is defined by using marker genes. So for example, stem cells in the bone marrow, they usually express CD34 or some other biological insights that allow you to do that. So in the epithelium, in the intestine, usually they are the LGR5 positive stem cells or some other stem cells that are also there. There's the BM1 stem cells as well. And from that, you then do the ordering or calculation of the pseudotime. In many cases, this is just what is the distance of every other cell in your data set to the original cell type. So for example, the original stem cell that you're marking, let's say the CD34 positive cells in the bone marrow. And that's basically just an overview of some of the methods that you can see. Monocle is one of them, but you see that like for T-scan, these are the decision processes that they were taken. So they used Euclidean distance, PCA, MCLUST, MST, and so on and so forth. This is a bit outdated version. There is no updated version of this either in the literature, but all of them actually follow this type of scheme or how the trajectory packages are usually suited. As I mentioned, one of the key components here that I was mentioning is creating this graph of ordering the cells. And that's usually done via MST, or that's what's called a minimum spanning tree. One of the first methods to define this method or using MST for trajectory is also monocle that I'll explain in a few slides. And what it does actually is just trying to find a set of points that you can connect using one graph so that the distance between them are minimized. So imagine that you have all these orange points around here, As you can see, for example, the connection between these 2 points, they are like, for example, to connect all these trees, you could, in theory, for example, if I eliminate this link, I could link this point here to the rest of the other ones by using this edge here. But as you can see, this edge has a higher value, so it's 9 instead of 8. So that will not minimize the distance once you sum, for example, like the 8, 2, 2, 2, 3, and 8. So this distance is what we are trying to minimize. So how can we connect all points using the least amount of edges, or edge values or weights that you have in here? So that's why in that way, you can actually try to find what are the relationship between these cells, so that from this cell, which cell can it go? It can go to any of the cells, basically, but this one is much more likely to be the next cell because their distance is much, much smaller. In this case, it's not that much smaller, so that there is a room for flexibility there. But so, for example, when you have this cell here compared to all the other cells, of course, the distance is much, much higher. So the likelihood of this cell being originated from this one or originating this one is much more likely because of this distance, which is much shorter. So these weights are usually the distance in your PCA space or ICA space or harmony space, or it can be a correlation of cells. So in this way, you can create a graph that connects basically all cells into one single trajectory, let's say. And this will kind of define your trajectories later on. One downside is that MSTs do not work in cell cycle. So if you have cell cycle in your data set, you cannot use this technique. And that's one of the reasons why sometimes you need to use another technique, actually, if you want to study only cell cycle. As I was mentioning, this was one of the first methods to derive this concept from MST. So basically, you have your cells, you can do dimensional anti-reduction, let's say PCA, harmony, and then you build an MST on this cell. So try to find a graph that connects all cells, minimizing their distances.
Sorry, do you have a question?
Hey, Paulo, yes, thank you very much. So you were mentioning, sorry, I just got stuck in the previous slide.
Okay.
That we try to minimize the algorithm try to minimize or find the minimal distance between cells.
Exactly.
But then, and you said that we had to do this in harmonized data, right? So, but then harmonized data already has eliminated variation. So good, like for gene expression analysis, we had to use the normalized data, not the harmonized data. Mm-hmm. But for use for that in order to avoid this integration and this variation reduction. But then here we try to find the minimal distance using data that has been already eliminated from that variation, right?
Correct. Yeah. The reason is because if you do not do that, then just like as for example, when you do clustering on your data set, you want to find clusters that are common across all your data sets. So let's say that's so that all T cells fall into the same cluster. Here's exactly the same. So we want the differences to be because of the cell differentiation process rather than by batch effect. Otherwise, you'll see, for example, the trajectory will start in one sample and then finish in another, even though those samples actually have exactly the same amount of, let's say, it's the same time point. So if the cells, for example, just like as in this paper here, is actually these are different time points, so different samples that were collected at different time points. But when you do the trajectory, you integrate all of them, so you put them into one big cohesive data set, and the differences will still be there. So like this one has very little neurons, and that's perfectly fine. But then these cells here will be just aligned into this part of the graph because they all match. As long as you have also connecting cells in here, as you can see here, you still have a few, they will always be part on that trajectory. So that the harmonization and removal of batch effect does not affect that much for your trajectories, and that's why you don't want to have batch effects on it as well.
But then
Exactly.
wouldn't it be the same for differential expression that you rather see the difference in data that has been removed of batch effect?
Exactly. So, but that comes of when you do the differential expression, you run the clusters. The clusters were identified on harmonized space. The same thing goes for the trajectory. The trajectories or the cell ordering is also done on the harmonized. But now, later on in my slides, I will show the differential expression on trajectories.
Okay.
Then you go back to the raw data.
Okay.
And then maybe that's where you're confusing. The differential expression on trajectories is also on the raw data, and then it's exactly the same.
I see. Okay, thanks.
Perfect. Yeah, so for monocle, you have your data, run dimensionality reduction and integration in that case, build a minimum spanning tree, and then you order the cells based on your biological knowledge, which cell is the one that is being originated first. So for example, here in this case, and then using the MST, you can kind of like identify this is cell type A, and then kind of a bifurcation, this is cell type B. And from that, you can then do differential gene expression across between the branches, between cell type 1 and cell type 2, and so on. However, one of the reasons why MST is not run on the raw cells anymore is because just running MST on all the cells themselves usually can be very misleading. So imagine this case here that you have an MST that was run on these cells, as you can see. But just by simply shifting one cell to the side, this tree can be completely changed in a way, so that the minimum graph that connects all these points are now this one. So you see that these graphs are completely different just by a simple shift in one cell type. And that's where Monocle 2 and other graph theories now, graph-based methods for trajectory inference are actually based. So what they do instead, they cluster the groups of cells. So they are not based on the individual cells, but they will be based on the clustering that you set. And that's why, like, when you do Slingshot, Paga, you do clustering, let's say using your Leiden clusters. And instead of using all the single cells, you use now the cluster centroids or some sort of metric between that like that represents your cell clusters. And then you run the MST on the clusters instead. And that usually is much more robust because if you change this cell in here, these cells are still part of the same cluster. The centroid will not change that much, and therefore the trajectory will still be a little bit more accurate than basing on a single cell specifically. And that comes to Monocle 2 and most of the trajectory methods that are out there right now. So it's exactly the same concept. So you have the raw data in a multidimensional space. You do dimensionality reduction, let's say PCA, and then integration, let's say harmony. From that, what it tries to do is then try to identify the cluster centroids around these cells, and then define a tree and project all these cells into this model. And that's very clear when, especially for Monocle 2, that you can see these cells being projected close to the model or to the MST, the graph tree that is connected. Monocle 2 itself later then tries to optimize this cyclically. So you have the model, and then you project the model to the raw data, and then you do exactly over and over. Other methods, they basically abandon a bit of this part of the modeling, the fitting. You don't need to know all of these details per se, but one thing that you will get as a result of that for all methods, you'll have then your cells projected into this minimum spanning tree or a graph that connects then all cells into one lineage. So there you can then select where is your start, and that is always a biased decision by you because you need to kind of know where the tree will start. These methods, they are not, they cannot do that for you. They cannot identify where to start, at least most of them. And I'll come to tell about a few exceptions on how that can be. Because the algorithms, they don't know biology, you need to kind of know where to start the trajectory. And from there, pseudo time is just calculated there is as the distance of each cell to the starting point that you set up. So then basically like that, this is the distance, and then this is the distance as colored there. It's just simply a distance to this original point. And in some cases, some algorithms, they already do also the branching automatically, but there is always a point where you can actually tweak that to actually fit your data better. And there's always like lots of parameters that you need to optimize to fit your model. So instead, many of these methods are still very biased to what knowledge and what model you want to know or do you want to extract from your data. As I was mentioning, there are a plethora of different trajectory methods out there. So in this course, we will explore a little bit on PAGA. And the idea of PAGA is very similar to the ones that I was just mentioning. So you have your graph itself, and you do then the clustering. As you can see here, you have, for example, your Leiden clusters, and then you compare the distance between those clusters, or like what is the probability of, or like the likelihood of one cell type connecting to the another, as measured as the number of connections that are between these clusters. And from that, you can then define trajectories. Where is your starting point, which is in this case is actually this green point here, and then you try to define the trajectories from there. Slingshot is the other one that you'll also explore a little bit in the R part, but it's exactly the same. So you have the cells order in a multidimensional space, let's say a harmony. You have the cluster centroids that are placed in here. And then from that, you try to project your cells into these graph and these lineages. And that's basically what you will do also in the exercises. There is other one that is basically based on Markov chains of trajectory as well. Cell router, very similar process as well. VIA is also used very similar to PAGA. You have a graph-based, and you also add a little bit of RNA velocity to try to infer where it starts. The same thing with cell path. You have a graph of connecting all the different cells, and you add RNA velocity, which I'll mention in the next slide, what it is, to try to infer where it starts and where it should end. And there are still many, many more, but in general, they are very similar in the structure. You have your data, you cluster it, and you then define a graph that connects all of them into one single trajectory. Do you have any other questions on this part before we go to RNA velocity?
Yes.
Please.
So all of these methods kind of rely on the fact that clusters that are near each other are going to be similar to each other.
Correct.
But then that's not always the case, right?
I would say yes and no. The reason like they rely on that is that because cells, they differentiate into these they don't simply jump into like in gene expression. They don't go from path A to path B or to cell B without having any cells in between.
Yeah.
So usually like the cells that are closer to them are usually probably the next path, right, to them. But there are a few exceptions, especially for example when you do data integration or analysis. Sometimes even the mature cells are very close together. So for example, if you work on actually T cells, so CD4 and CD8 T cells, they are always like, because they are so similar to each other, there are only a few genes that actually differentiate. Mm-hmm. So when you do dimensional length reduction, they are always close together. But it doesn't mean that they one originate the other. But actually there might be other progenitors that actually sometimes, for example, if you're not looking at the bone marrow, if you're looking just at the blood, you only have the mature one. So you would assume that maybe one goes to another, but that actually doesn't happen. So that's in some cases that might not still be the whole true. It's just because the cells that let me reformulate that. Is that sometimes the progenitor cells are not in your data set. And then cells that it appears to be different, let's say CD4 and CD8 T cells, mm-hmm. They will just fall together just because you don't have the progenitors. If you would have the progenitors, then they probably will be a little bit more far apart, let's say, in your embedding. And yeah.
But does that mean that this, for example, now if I see the image of cell router,
Yeah.
like what I see there, exactly. So what I see there is like there are these separate clusters
Yeah.
that we can find the identity of course.
Yeah.
And then because of our biology knowledge, we can determine that, yes, this is bifurcating into these two and so on, right? But what
Exactly.
if, for example, our UMAP doesn't really follow that?
Yeah, so that is also an indicator. Of course, the UMAP is what I would not advise to run trajectory. So you will run trajectory on the raw space, and that's why not using the UMAP or the TSNE that much. But they do provide, as you're mentioning now, they do provide some guidance that maybe these cells shouldn't be put together, right? And it's really like an investigative approach. You always need to check and see, because you can basically fit anything into trajectories if you want. Even things that,
Yeah, exactly.
Yeah, so I see your point, and you're definitely absolutely correct on that. Especially, for example, in this case, let's say that they are not connected. You probably don't have even the intermediate, but
Yeah.
then you're trying to fit trajectories onto cells that actually don't make any sense. Let's say, oh, I want the neutrophils to give origin to the T cells because they are closing space for some reason. Yeah, that does not make sense. So be always careful when fitting the trajectories. And perfect note there from you.
Yeah.
Thank you.
Thanks.
All
Can I
right.
ask something also?
Yes, please.
Sorry. I was wondering, you use your Seurat cluster in Arden, you use your Seurat clusters, right? So then this also really depends on how much clusters you say that you want to have there. Like if you
Correct.
if you have like only a few clusters, then it could be that then cells that are maybe precursors of something are in the same cluster, right?
Yeah.
As the cells that so how would you then decide kind of what is
Yeah. So for
the right
trajectories,
amount?
so for trajectories, what I would recommend is always to try to do over clustering. So for example, sometimes when you do clustering, let's say that you have these trajectories and so on, if you just do clustering by itself, you might get, for example, like a lot of these cells into one big cluster. And then you won't see these like very tight differences like between, or the gradient differences. Of course, when you do over clustering, sometimes what you don't see much difference between the clusters, like let's say this cluster versus the one before, because they are still very close. But for trajectory is actually good. I mean, you do the general clustering just to see, this is this cell type, this is that cell type, and then you kind of see that there are some transitional cells in between. But for then running trajectory, it's usually advisable to do the overclustering, and we'll see that on the exercises a bit as well, that you don't want just to separate these ones into digestible chunks so that when you connect them, then they will be like a cluster 1, cluster 2, cluster 3, and then you see the clear path between them. Yeah, I hope that answers a great question.
Yes, thanks. Great.
Yeah, so just recapitulate. It is okay to do another round of clustering on your Seurat or ScanPy object just to run trajectory on, so that you get a better depiction of the trajectory lineages. Yeah. And that comes also now for another method, and this is actually one of the coolest methods in terms of trajectory that there is. And this is called RNA velocity. So what is RNA velocity and what is based on? So let's step back onto the RNA dogma. So there is here I'm showing the RNA maturation and how we quantify that in single cell RNA sequencing. Imagine that you have done your DNA, and once that DNA starts to be transcribed, it produces what we call a nascent DNA or an unspliced DNA or RNA, sorry, nascent RNA or unspliced RNA, where you have exon. So here I'm coloring the exons just in different colors for illustration, but these are the same genes. And then you have the intron that is in between that usually is not does not produce a protein. Once that nascent RNA is pushed out of the nucleus, there will be a process that is called splicing, where you remove the intron, and that's probably Biology 101, and then the two exons will be then joined together. One thing that you can actually do to try to quantify these two types of RNA separately is by then trying to quantify where these reads, once you do the sequencing, where do align. But one thing that you should always keep in mind that is the nascent RNA is the one that is produced before, right? Because it's just being transcribed after DNA and is always transcribed with the intron. And then after that, that RNA will be mature. So keep in mind that then all the variations in the nascent RNA or the unspliced are followed by the variations on the mature one. So that means if you see, for example, a lot of differences in the nascent RNA, that means that this RNA is just being transcribed. It's just a process that just started in that cell, compared to mature RNA, which have been in the cell for a little bit longer. And you will see that in a few examples later on. But one thing that was noted by Sten Linnarsson's lab at Karolinska, where this paper comes from, is that you can actually then quantify that in your experiment at the single cell level. So here is just an example. Let's say that you have your mRNA in here, and then each one will be a read that you see in your Illumina sequencing. So you have the reads that span either the exon or the second exon or the intron, or in here you have the mature RNA. You can see where the reads align to both of these exons. And what you can quantify, and that's what Velocito program will run on your data set, is then not only just extract the reads, but map them into 3 different categories. One that is all reads that are in a mature RNA, and that means because the mature RNA have exon-exon junctions, you can see that the reads that are mapped to this matrix will be only the reads that are present in mature RNA. The matrix that contain the nascent or the unspliced reads will be mapped to this matrix because they contain parts of the intron that were still not removed. So this is RNA that hasn't been spliced yet. And then all the other reads that map to either of them will be projected and counted into this ambiguous matrix. And this is what if you run Velocito, you'll get exactly these 3 matrices. And here I'm just illustrating what these matrices contain. And what you do in RNA velocity, actually, so Stan's group, they also went a bit further, and they start modeling that into biology and using that computationally to actually infer where cells are going from that. So here, again, it's just the same statement. So the unspliced are followed by the spliced. So the nascent is then followed by the mature RNA. Here is exactly the same thing as I mentioned. So the DNA is transcribed into the unspliced mRNA, then you have a splicing, and then you have the spliced mature RNA, and that mRNA later is degraded. You can follow, for example, a cell. So for example, here is a cell that does not express any RNA of gene A, let's say interferon gamma. So once the interferon gamma gene is upregulated, what you will see is an increase on the unspliced version of the RNA first, and that later is being followed by the spliced version of it. So you have more activation or induction of expression once this gene is in this area. Once it reaches this state, this is a state where the amount of RNA that is being produced and degraded is the same. So the cell is basically activated and producing interferon as much as it can. And that is what we call the steady state of that cell. And another thing that can happen is that now this cell is producing interferon gamma because it's activated or something, and then maybe it can be silenced by, let's say, a regulatory T cells, then shuts down that signaling pathway. When that signaling pathway shuts down, this cell will then reduce the amount of unspliced because it's not producing our interferon gamma anymore. And the amount of splice is still high, but it's slowly decreasing because the mature RNA is being now degraded. And then this cell basically goes back to not producing interferon gamma anymore. And this is what you'll see in many velocity graphs. And basically is this concept of induction and regression of mRNA production in a cell. So looking, for example, in the example of a circadian gene, here you can see the spliced version and the unspliced. of the same condition of the same cell. And here is just a condition afterwards. But one thing that you can notice, for example, I'm showing in the arrows here, is that the unspliced or the nascent RNA is much closer in space, like you see the profile here, is very closer to the spliced in the next time point, meaning that the nascent RNA is basically predicting the next stage of the mature RNA in the next time point. The same thing happens for here. You can see that the unspliced or the nascent RNA is very similar to the mature RNA in the next time point, and so on and so forth. And that then allows you to try to measure, oh, I have the unspliced and spliced counts within a cell. And then that allows you then to project, oh, this cell type is probably differentiated into another cell type that I can also see, which is another cell, another time point. Let me put you that into a visualization tool to make it easier. And this is also developed in the same paper as well, the RNA velocity vector field. So what it shows here is that the RNA velocity allows to do then create an arrow, like a direction to where the cells are going transcriptionally based on the spliced and unspliced versions of the RNA. It helps you also define the starts and ends and probably a few bifurcations as well. So you can see in this UMAP, you can assign these vector fields. They are pointing to some areas of the plot. And you can see that where basically they are going. And that allows you to not only identify which are the most terminal states. So for example, if you reach these points in here, let me just move this arrow, this box in here. You can see where the diffusion is trying to end. So for example, you can see here that the arrows are always pointing towards there, and then they reach the end. And that's basically what is being captured here. So these are the diffusion endpoints. The same thing, there's like a back diffusion, or where the diffusion is starting. And that's basically here. And because of that, that allows you to then identify where are the progenitor cells and where are the fully mature differentiated cells in your data. So that's instead of the previous algorithms that I was mentioning, that you need to know the biology to set where is the starting point. In here, you can define that by the data itself. As I was just mentioning, the arrows then point to the position of the future state or the spliced version of the cell. So these are kind of extrapolated from the velocity estimates. So if you have, for example, an unspliced cell in here, you try to find where is the cell spliced in your multidimensional space, and then you can draw an arrow between them. And basically that's what the vector fields are trying also to point out in here. So if you have a cell in here, where is the next mature RNA version of this cell? And it's up in here, so that's why you draw an arrow in that direction. There are multiple flavors of RNA velocity. There are many packages that came out of the same concept. So it's just updates of that same package. So for example, SCVelo by Fabian Thai's lab introduces this dynamic modeling. Sometimes the velocities are kind of pointing to the wrong directions. So it tries to adjust that for a few examples. So you have a dynamical model where you have here cells that usually are not migrating to any or not being differentiating to any point, but rather only the cells that are really in between the two clusters will kind of have a trajectory point. SDVelo also tries not to force velocity in control cell state, let's say progenitor cells that are actually not being differentiated. So they try to update that model. There is also another model for cell cycle that is a little bit improved. And there are still many, many others that came after that. So choose the best, or just simply go, maybe SCVelo is actually quite user-friendly, or Velocito from Lina Schoen is also really good. Any questions on this part? If not, then I'll go to just a few hallmarks into other packages that are being developed at the moment. So there is now this cell rank 2. Maybe also later we'll give a brief on her experience on that. But basically, what it tries to do is now combine multiple of these types of analysis. So, for example, you combine RNA velocity with the clustering of the cells and their similarity, with pseudotime, with maybe even additional markers of maturity of cells. Maybe you have experiments with different time points, so you know that the cells are differentiating. Or if you have some other cell type labeling, let's say lineage tracing, and you can try to combine all of that into one big trajectory that tries to find where is the initial state, where is the terminal state. So these are more like advanced techniques. I haven't used those yet. But those seems to be also very promising to try to integrate multiple clues from different types of sources or from your data into one more accurate model for trajectory. The same goes, for example, now this Moscow, which is exactly very similar to that as well. So it tries to find clues from either even spatial time, experimental design, let's say different time points, and trying to match cells between different conditions and create one big model that allows you to then model how cells differentiate from A to B. And yeah, this one is very, very, very new as well this year. And that brings us now to the last part of this lecture, which is differential gene expression along trajectories. Coming back to your question, where should you do differential expression now? Now I would recommend that taking the raw data, like the raw counts that of course, you can use the normalized or the raw counts depending on the method that you use. But definitely you should use that on the not batch corrected counts from your data set. Let's start simple, because differential gene expression within trajectories can become complicated very, very fast. Even if you have like multiple lineages, multiple trajectories, it can become very, very complex in a minute. But let's take just an example. You have the hematopoietic stem cells that differentiate into the red blood cells in your blood, or neutrophils. So imagine this gene, for example, here that is being upregulated on the ends of the trajectory in here. As you can also plot as an expression versus pseudotime. So the pseudotime is the distance between these cells to the hematopoietic stem cells. And here I'm just highlighting at the clusters just so you have a visual guideline where they are. And you can see they are very, very similar. And there are a few different tests that you can do. So one is called an association test. So you just see within the orange, so you just ignore the blue. Within the orange, are there any differences within these time points? As you can see, there are like the time points are changing and increasing over time. So then it's kind of creating a ANOVA test that you just try to find, is there any differences in here? So you use all the time points to do a differential expression test. And in this case, it is differentially expressed. Another test is just comparing the start versus the end. Sometimes there are a few curves that they will be upregulated and then they go back. But in this case here, you can see that the final endpoint and the start endpoint, they are different. So this test will give, for this test, this gene here is differentially expressed because the start and the end are different. If you take now another example, another gene, let's say that you have a gene that is usually not expressed in the orange, but is only upregulated in the blue. This is how, let's say, if you plot the gene expression versus pseudotime would look like. The blue is upregulated, but for the orange one, it's not. So if you do an association test, you do basically an ANOVA on all time points and see if there is any gene that is being, or any signal that is being captured here, and you will see that it's not differentially expressed. And if you compare the start versus the end on the start versus end test, it's also not differentially expressed, and then this will also not be differentially expressed in this test. Now, if you go to comparing between the lineages. So it's exactly the same lineages in here as well. So I just go back and forth. So you see this is exactly the same. But now there are three other tests that you can actually use to compare. So for example, differential end test. In that case, what you're comparing is that you're comparing the last point for both of the lineages. So you're comparing the green versus the blue one. And if you can see, they are very similar, so they are not differentially expressed. When you have the pattern test, you're actually testing all the different time points together. And you can see it's not differentially expressed. All of them are very similar. And early D test, you're actually testing not the start point, but just after that, because then you're just wanting to see if there is any difference that is in the beginning of the trajectories that is significant between the two lineages. In this case, it's also not differentially expressed because they are very similar. The same thing goes for example, if you have this gene in here, you see that the blue one is upregulated, the orange one is not. If you just do a differential end test, you're comparing only the last point, and then you can see they are differentially expressed. If you have all the time points together, you can see if they are differentially expressed or not. And the early time point is also comparing only these ones, and you can see kind of they are differentially expressed. This is just a very simplified view, an example of how these tests are being performed. But if you go, you can actually, I wrote them down as well here later for you to take a look and think, oh, this is kind of a student t-test between condition A and B, or it's like an ANOVA test. This is basically what I just explained to you, but just written so you also can dive deeper a little bit later. And our recommendation for running differential gene expression on trajectories is use this package, which is called Trajectories, and that's what we are going to use in the R session. There is now a Python version that was just released as well, so we'll update these now in the next course because this is quite new as well. But it's basically like implementing these types of differential expression tests on trajectories for Python. There are a few tests. So for example, for the Python version, we're using the internal PAGA trajectory and differential gene expressions, but they are very limited as they are. And some other tools also offer differential gene expression, but they are still also somewhat limited. In this Trajectories package, actually, they also mention like additional examples. So I just show you these first two, but there are other examples that you can kind of see if it is differentially expressed or not. But at least with the examples I showed below before, you can kind of identify why they are not differentially expressed or not. Any questions on this? And I'm almost done on the lecture now as well.
I just wanted to ask a question about, like, suppose you have a module of genes, which like some of it will be like going through this trajectory from cluster 1 to cluster 3, maybe up, and then there is like another subset of genes in the same module which goes down. Do we have to run like the differential gene expression analysis per gene, or does TradeSec and they can integrated by itself, like this subset goes down in this trajectory and this subset goes up, and it can do it by itself, or do we have to run it one by one?
Excellent question. So by default, TradeSeq will do that at per gene level. So you, like when you do the test, also you will choose which test you're running and why you're running. But actually, given your point, so for example, if you have the gene modules, what you can actually do then run the WGCNA or some other type of gene module identification to group the genes and then run differential expression on those. That should be still fine. As well, I think it will give you kind of the same results as well. It's perfectly fine to use. But by default, it's run on the raw gene expression. So like on the per gene basis.
Okay, thank you.
You're welcome. Great question. Yeah, so to wrap up, one of the things that I was mentioning just in the beginning, can you simplify your trajectories into more simple structures or topologies? try always to do that. Subset your data set and run trajectory only on the things that are interesting to you, rather than being worried about fitting trajectory on cell types that are not even related to what you're looking at. But here in this paper from Salens, you can actually see like a decision tree. So do you expect multiple disconnected trajectories? Like, are you trying to model many cell types together? If yes, then try to use a disconnected method. And I'll come to the methods in the next slide. If you don't expect to do that, for example, maybe you subset only the epithelial cells, and then you go with that. Do you expect a particular topology? So for example, is a cell cycle that you're trying to evaluate? Is it a linear trajectory or a bifurcation or a multifurcation? Like in the bone marrow, you probably will go through either of these ones, or a little bit on the trees as well. If you do expect cycles in the topology, and it's kind of a complex topology, like for the embryonic neuroprogenitor cells, that it has like the cycle and then differentiating into other cells. If yes, then you probably should go to a graph topology. If you don't expect cycles, then you can just go for like a tree topology that is basically one of those two. They also have like an interactive browser that you can actually check. And that's basically this table here is also on their paper. So what they did was compare many different trajectory inference methods, checking like the priors, if you need to know, have like some biological knowledge on information to do trajectory analysis, which platform, if it's R or Python, and the topologies that I was mentioning. So that's why it's important for you to know which biological process are you trying to depict, because that really depends on which method you can choose. Here I'm actually just cut the most common ones. There are a few other ones that, for example, only look at cell cycle. There are others as well in the paper. But here you can see Slingshot is one of the best, at least in terms of overall accuracy, and that's why we chose that one. And also for Paga in Python is also one of the best, so that's why we chose those to teach in this course. There is different variations of that as well. As you can see, that's Monocle 1, Monocle 2 is also here, that I also mentioned, but they are being in less use nowadays. Slingshot and Paga are very flexible and allow you to do a lot, and that's why we brought those for you today. So please check out this paper or also their interactive tool. Actually, it's kind of still a copy of the paper, but just interactive if you want to check. Otherwise, as I do as well, I still follow all the instructions in this course when I do want to do trajectory, because they're just so useful. And my final considerations. In your data set, when you're trying to fit these trajectories, make sure that you have a trajectory. Let's say that you have T cells and B cells in your data set, but you don't have any of the progenitors before them. So does it make sense to run a trajectory there? Maybe not. So you can simply do not do that. Just follow the traditional differential gene expression between clusters, and that should be more than enough as well. The trajectory is better once you have all the intermediate cell states. So make sure that you have all of them. So for example, if you have B cells and T cells, of course, in the peripheral blood, you don't have the progenitor of those if you want to find the trajectory between these cells. So you need to look at the bone marrow, for example, where you have the intermediate states within that gives rise to these cell types. So make sure that you always have intermediate states in your data set. Otherwise, you can really fit any trajectory in your data set. Make sure that you know if you have any branching in your trajectories, and that's basically why you can define if you're like, do we have a bifurcation, or if it's a linear trajectory. It is important for you to kind of understand. You can also see that from your data itself. Sometimes when you do a UMAP or a visualization of the graph, you kind of see if there is a branch in there. So make sure to account that in your method that you're using for trajectory. You can, as I was mentioning also in the previous slides, if possible, feel free to subset your data set into smaller trajectories that are easier to understand and easier to do analysis. So creating these multi-connected graphs and try to fit trajectories in this is much harder than simply do a bifurcation trajectory. So you definitely can subset your data set and run trajectory on specific parts that makes more sense and it's easy to understand. And as always, be aware any data set can be forced in trajectory without any biological meaning. Let's say I have two different clusters, B cells and T cells, on mature blood, on peripheral blood. In theory, you can see trajectories there, but they don't mean anything. So be aware that that can happen, and don't do that. So try to be make sure that you have all the intermediate cell states, for example, in your data set. And also another important consideration is to make sure that the genes that you use for dimensionality reduction, let's say the harmony, are capturing that what you expect in terms of differential trajectory. Let's say that you want to do differential expression or trajectory within T cells, but you remove all the T cell markers in your harmony. So that actually happens once you select parts of the dataset. Let's say that you have the dataset on the bone marrow and you want to create trajectories on the T cells. But what you need to do is then you take the T cells, run harmony integration, run feature selection again, and then run trajectory on these, not on the gene features, highly variable features that you had for the whole dataset, because that also includes genes, let's say neutrophils, B cells, and other things. So take the T cells specific that you want to run trajectory, then run highly variable gene selection, scaling the data, harmony integration, and then you basically will create a trajectory that only fits the T cells. And then once you draw the trajectory on these, they will much more recapitulate biology rather than artifacts because of selection of the genes. And that's what I had for today. Thank you so much for listening and for excellent questions as well. I'll be happy to take any questions now, and I'll stay a little bit longer also for the courses. I'll be there if you want to also ask any other questions. Thank you so much for the invitation, and I hope you enjoyed it.
Thank you, Paulo. Super nice. Do you guys have any questions?
Yes, thank you, Paulo. This is really interesting. I have a question about the trajectory topology right now. If we know for a certain process that it's kind of reversible, it's not cyclic, okay, not the cell cycle, but it's a process that should go linear if everything goes well. However, it can actually revert back in a cyclic way. In that way, what would be the ideal topology to study here?
That is a great question, and it's a very hard problem as well to solve. Let's imagine, just give an example of a process that happens in that case. Let's say T cells. So T cells sometimes differentiate into, for example, Th17, but then these Th17 can actually differentiate into Th1s or so on, or actually be what we call the XTH17. So they can express other markers, so they are actually very plastic, so that they can transition into different differentiation states. I don't think there are like many tools that are able to handle that. So what you need to do is then select, for example, the Th17s to Th1 and try to just select those cells and then do a linear trajectory on that. Because then you kind of can try to see what are the paths between cell state 17 to Th1s. Because most of these, they are kind of assuming that there will be always an end. So these kind of reverse process is usually, as you can see in all the arrows here, they are usually never taken into account. Unless you have kind of a cyclic pattern, but I haven't seen that yet, at least, that you have like Th17, and then there's a state, and then there's another one that is Th1 maybe, and then there's another state. But I have never seen these cycles into T-cell plasticity. Usually it's just a direct link between them.
Yes, because I was thinking of the connected graph as a way of, this intermediate cycling. Then the intermediate cycling can either, after a certain point, can either go to either direction.
Exactly. So it is a very hard problem to solve, I think. I don't think if any computational tool is able to handle that. So the only way that I would see is then subsetting your data for cells that you think are transitioning from one to another, and then try to run the linear.
Yes.
Yeah.
Okay. Thank you so much.
Yeah. Thank you. Great question.
Thanks so much. Super nice. I was wondering if you have then from one sample, you have different time points, different biological time points. How would you kind of integrate that in your trajectory analysis? Can you do that? Like,
Yes. So I think that comes back also, for example, in this example that you have, for example,
yeah,
10.6
okay.
that you, so
Yeah.
that is kind of solved on when you do the integration of your data set, right? So
Okay.
like these cells here, for example, it doesn't have not mature neurons at this
Mm.
stage, but in the other stages it has. So that's why when you run the trajectory, the harmony in space that you're running trajectory or the clusters that you're defining are always in the integrated space, so you remove these batch effects. But later, when you do differential gene expression, you're not accounting for the biological time. You can always plot versus the biological time, but you will see more into what is the state of each cell. So like even though this one doesn't have mature neurons, but when you do the trajectory, you know that these ones are the ones that are more mature. But you can kind of see this gradient between the neuroblasts versus the mature neurons in the end.
I see. Okay, thanks a lot. Yes.
I think it can also be really helpful when you have time point to sort of define the end points and starting point of your trajectories, right? Where you have more of the early time point versus more of the late time points. To
Perfect,
incorporate
exactly.
that into.
Yeah. As you can see here, for example, some of the most early progenitors are much more abundant compared to the mature ones. So you can kind of see, okay, this is definitely a mature state, especially for this system where you kind of know that. Any more questions?
Thank you.
I have one question, but
yes,
maybe it's a little bit naive. But like for this RNA velocity, so what would happen if you actually have a problem with the splicing? Because you assume the splicing is working and then you can draw the trajectory based on that. But if it's not, if it's aberrant in your model, like...
Yeah, that is a great question. So most of these packages, they try to, so this is the first model. So it was a quite simplistic one. Other types of tools, they try to model that a little bit differently as well. So adding, for example, other types of degradation rates or even microRNAs or trying to inhibit with siRNAs, because as you mentioned, this process is not always straightforward, as you mentioned. So like some genes have this splicing pattern happening all the time. Some have that deactivated for some reason because of like a dicer problem and so on. So definitely, so I don't think any of these methods can actually handle many of these complex tasks. But in theory, if you have a problem on the dicer, so if you have maybe like say a control that you have a normal splicing machinery versus another one that you have a problem, maybe that's a control knockout mouse, you probably will see a difference in this plot here. When you have like, for example, the amount of spliced and unspliced, they might not even be different, or they might be completely different. You never will see the spliced version anymore. But definitely you probably will see in this plot, I think, if you have a problem. But these methods do not take that into account. No.
Okay, thank you.
I could even be wrong because maybe I haven't checked exactly the details on every single one of those. These are just the overview. There are a lot more. Maybe there are a few that can do that, but I'm not aware of it.
I think it's probably far too complex to model, right? Also, if you have a few genes that deviate, that have like aberrant splicing, it's still the joint signal from multiple genes that makes you then draw these sort of arrows with velocities. So hopefully it should be fine.
Yeah.
Should also be mentioned that with nuclei data, it's not a very good idea to try to do this velocity since we don't have a large proportion of the spliced.
Correct. That is a great point. Yeah, so most of, like I was mentioning in the lecture, most of the nascent ones are still within the nucleus because they're just being transcribed. And the splicing usually, at least most cases, occur on the cytoplasm, so they need to then transition out of the nucleus. So if you have data sets that only have the nucleus part, you're more likely to only have nascent RNA. so the concentration of nascent RNA is much, much higher than the rest. And so that's why, like, oh, you don't see the mature RNA. It doesn't mean that it's not there. It's just that in your method, you're just not capturing it. Perfect observation also.